So in this video we are going to see about subcutaneous fat. Embryology, subcutaneous fat is derived from mesenchyme like the dermis, and these fat cells they start developing during the end of 5th month of intrauterine life. So these adipocytes nothing but fat cells. They synthesize so much fat in them that they push the nucleus to the periphery and they give a signet ring shaped appearance. So the basic unit of subcutaneous fat is this primary microlobule. So this is a primary microlobule, and this primary microlobule is formed by collection of a number of adipocytes. This is about 1 micrometer in diameter. So this primary microlobule they aggregate to form a secondary lobule. So this is a secondary lobule and this is about 1 centimeter in diameter. So if you see, this is the fat and they are separated by fibrous septa running between them. So from this fibrous septa we have collagen bundles running through and separating the primary lobule. So this is the secondary lobule and inside you have many primary lobule, and these collagen bundles they run and they surround each primary lobule also. So this portion of subcutaneous fat that projects upwards into dermis, you call it as papillary adiposae. So this papillary adiposae, it surrounds the hair follicles and then the eccrine glands and also blood vessels. So as I said, this fibrous septa, we have collagen bundles running from the fibrous septa and they surround each primary microlobule. It is not seen through routine staining and hence you need a reticulum stain to visualize that. So this interstitium in subcutaneous fat, it accounts for about 10%, whereas in a normal tissue, the interstitium is about 20%. So it contains few fibrocytes, mast cells, mucopolysaccharides, collagen fibers, and free nerve fibers. So this is the blood supply of subcutaneous fat. So these large blood vessels, they run in the fibrous septa, and from these large blood vessel, if you see the arterial supply, a branch of artery, it supplies the secondary lobule. From that, a central arteriole arises and it reaches the primary lobule. And regarding the venous drainage, they always start from the surface in contrast to artery which reaches the center. So there is an intricating network of capillaries between the central arteriole and peripheral venule. So these large blood vessels, they also supply the dermis and the subcutaneous fat. But the network of capillaries, there is no capillary connection between the dermis and subcutaneous fat. Dermis and subcutaneous fat, they share same large blood vessels, but capillaries are different. So the fibrous septa, they also contain lymphatics and nerves, and the subcutaneous fat, they surround the secretory tubules of sweat glands and lower parts of hair follicle. So the adipocytes in skin, by the action of lipolysis, they produce free fatty acids and glycerol, which they need to be sent to general circulation. So for this process, they must be located close to the capillaries that contains properties like high permeability, low hydrostatic pressure, and shortest distance. So the distribution of fat is of two types. One is gynoid distribution and other one is android distribution. So this gynoid distribution is usually seen in women and it is situated mostly, the distribution of fat is like breast, buttocks, hips, anterior thighs, inner aspect of knees, lower abdomen and pubic region. Whereas the android distribution is mostly seen in men and the distribution of fat will be in the nape of neck and then the epigastric, that is the abdominal region, and then the deltoid region. So in both sexes, however, there are certain sites that contain less amount of fat, includes the eyelids, earlobes, scalp, dorsum of hands and feet, nostrils, scrotum, penis and clitoris. So as I mentioned before, this papillary adiposae is the projection of adipose tissue into the dermis. So this is the adipocyte. You can see it contains organs like mitochondria, Golgi apparatus and nucleus, and this central large area is the fat reservoir. And also you can notice the signet ring shape. So the function of subcutaneous fat, it acts as shock absorber to protect the vital organs and it helps in the mobility of skin. It helps the movement of skin over the underlying structures, and based on the gynoid and android distribution, it gives a cosmetic appearance and it helps in thermoregulation and acts as an insulator for heat, and it mainly acts as an energy storage area which comprises 90% of triglycerides and it acts as receptors for neurotransmitters and hormones. So it has both alpha and beta receptors. These beta receptors on stimulation they promote lipolysis and production of free fatty acids, whereas alpha receptors they inhibit lipolysis, and the free circulating hormones that promote lipolysis are glucagon, growth hormone, thyroxine, ACTH, and catecholamines. They also secrete leptin and interleukin 6, and they convert androstenedione to estrone, which is a major source of estrogen in men. So this adipocytes, and this is the signet ring shape in adipocytes. You can see they have synthesized so much fat and pushed the nucleus to the periphery. So in routine processing, the adipocytes they appear as empty spaces within the cells and are not visible. So to make them visible, you need glutaraldehyde fixed or plastic embedded space. It contains one large central lipid droplet and in histopathology, the young fat cells they contain two or more small lipid droplets, whereas the mature fat cells, as seen in the picture, peripheral nucleus. So applied aspects in scleroderma, the subcutaneous fat will be replaced by dense fibrous connective tissue, and in Werner's syndrome, there is absence of subcutaneous fat in the areas where the lesion is present. So the special stains include vimentin that is non-specific S100. It stains the mature adipocytes, and calretinine for lipoma and liposarcoma, and then oil red O and Sudan black for neutral fat. This Nile blue sulfate it stains the neutral fat pink red, whereas the free fatty acids blue.